Welcome to part 3 of our 4-lecture series on clinical genetic testing. This lecture will focus on single gene testing, multi-gene panels, and whole exome and whole genome sequencing. On this slide, we're using sentences to help visualize how different types of genetic variations affect our DNA. The original sentence is, "The gray cat ran down the hall," which represents a normal sequence. Now let's look at a sequence variant. Here the sentence changes to, "The cat ran down the ball." A single word is altered, just like how a single base pair change can occur in a genetic sequence. These kinds of changes are typically identified through sequencing methods like Sanger sequencing and next-generation sequencing, which give us a detailed readout of the DNA sequence. Now we have a deletion. The gray blank ran down the hall. You'll notice a word is missing, just like a genetic deletion removes a part of the DNA sequence. In a duplication, we see the gray cat ran down the hall, where a word is repeated. This represents a section of DNA being duplicated. To detect deletions as well as duplications, we use methods like multiplex ligation-dependent probe amplification, array comparative genomic hybridization, and next-gen sequencing. Now we move to sequence variants that might change the meaning of the sentence, but in ways that aren't immediately harmful or helpful. For example, the gray cat ran down the mall. Here, the meaning is slightly changed, much like how a variant in our DNA may change a gene's function without causing disease. Finally, we have something called a variant of uncertain significance, or a VUS. In the sentence, the gray cat ran down the stall, the change is different, but it's unclear if this alteration affects the overall meaning or function. In genetics, a VUS is a variant we can't yet classify as harmful or benign. These different types of variants can help us understand how mutations impact genes and how we detect them using various methods. Here we compare sequencing to a spell checker for a gene. Just as a spell checker scans for errors in your writing, sequencing analyzes the DNA sequence of a gene to identify any mutations. Just like in our example, where cat doesn't alter the overall meaning of the sentence, but introduces uncertainty about its correctness, VUSs represent genetic variants that are not clearly classified as either pathogenic or benign. They can arise from various genetic tests and create challenges in interpretation, leaving both patients and providers seeking clarity. Understanding VUS is crucial as we navigate the complexities of genetic information. It highlights the need for ongoing research and collaboration in the field of genetics to better interpret these uncertain findings and provide meaningful guidance to patients and families. VUSs can occur with several different types of tests, including karyotypes, chromosomal microarrays, sequencing, and even non-genetic tests, including imaging techniques like MRIs, CT scans, and ultrasounds that can also reveal findings that lead to full variants of unknown significance. For example, let's consider the example of an echogenic intracardiac focus, or EIF. This is a small bright spot seen in a baby's heart during an ultrasound exam. It's thought to represent small deposits of calcium, also known as mineralization, in the heart muscle. EIFs are quite common, found in about 3 to 5% of normal pregnancies, and they do not cause any health problems. However, because they are observed without clear implications, they are classified as a variant of unknown significance. On this slide, we're comparing genotyping and next-generation sequencing, or NGS. Let's start with genotyping. This method is commonly used in carrier screening and direct-to-consumer testing. It focuses on identifying common mutations, particularly those that are well established and associated with specific genetic conditions. Genotyping often takes into account the individual's ethnicity because certain mutations are more prevalent in specific populations. In contrast, next-generation sequencing, or NGS, offers a more comprehensive evaluation of genes. NGS allows us to analyze a broader range of genetic variations, including small insertions and deletions, also known as indels. However, it's important to note that while NGS is excellent at picking up these small changes, it may not identify larger indels. Additionally, NGS is ethnicity independent, making it a powerful tool for individuals from diverse backgrounds. However, it does carry the possibility of discovering variants of unknown significance, or VUS. On this slide, we're comparing the Sanger sequencing method with next-gen sequencing. The Sanger method, which was developed back in 1977, sequences a single DNA fragment at a time. This traditional approach is highly accurate, achieving a remarkable accuracy rate of 99.99%. Because of this accuracy, clinical laboratories often use Sanger sequencing to validate gene variants that have been identified through NGS. In contrast, NGS is a game changer in genomic testing. It allows us to sequence millions of DNA fragments simultaneously in what's known as a massively parallel approach. This capability enables us to capture vast amounts of genetic information in a single run, making NGS an invaluable tool in both research and clinical settings. Despite its advantages, Sanger sequencing remains popular for specific applications, particularly when sequencing a short region of DNA across a small number of samples. This method can accurately read up to 1,000 base pairs without the need for complex data analysis, making it straightforward and effective for focused genetic testing. Now let's take a look at single gene testing. Single gene testing has benefits and limitations. Single gene testing is highly targeted, making it ideal when there is a known familial history or when a specific syndrome is strongly suspected. This can be especially helpful when the clinical presentation points to a particular condition, allowing for a precise and focused approach. Another advantage is that single gene tests tend to be less expensive compared to broader tests, like whole exome or genome sequencing. Additionally, because the testing is so specific, there are usually fewer opportunities for variants of uncertain significance, which simplifies the interpretation and discussion of results. However, single gene testing has its limitations. It may not be the best choice for complex disorders that involve multiple genes or factors such as polygenic conditions. And often, if the test doesn't yield informative results, you're left asking, what's next? You might need to follow up with broader genetic tests like panels or exome sequencing to get the answers you need. In summary, single gene testing is powerful when you know what you're looking for, but it can fall short when the genetic picture is more complex. Some applied questions that I want you guys to be thinking about in terms of single gene analysis include, on the single gene level, what does sequencing miss? How about deletions and duplications? In what instance would you not automatically order both modalities? And if there is a known familial mutation, what is the best testing approach? Feel free to pause the video here and jot down your ideas. Let's shift gears from single gene testing to gene panel testing. Multi-gene panels offer a broader approach to genetic testing. However, there are some benefits and limitations to keep in mind. With multi-gene panels, we're essentially casting a wider net, testing multiple genes at once. This is particularly useful when a disorder could be caused by mutations in any of several genes, allowing for a more comprehensive analysis. Another key benefit is that multi-gene panels typically have high detection rates, especially when used for specific disorders or syndromes where the genes are well known. However, this approach comes with some trade-offs. One of the main challenges is the increased likelihood of finding variants of uncertain significance or incidental findings, which are expected results that may not be directly related to the patient's clinical presentation. This can complicate counseling and management as not all findings are immediately actionable or understood. Now let's explore when you might consider ordering a multi-gene panel. First, a panel is useful when there's a suspicion of a genetic syndrome that's associated with multiple genes. A great example of this is Noonan syndrome, where mutations in several different genes can lead to similar clinical features. Secondly, multi-gene panels are helpful when there's a suspicion of a genetic syndrome with overlapping clinical features. This is often the case in conditions like cancers, ciliopathies, or connective tissue disorders, where the clinical presentation could be linked to a variety of different genes. The panel allows us to cast a wider net to ensure no potential genetic causes are missed. In these scenarios, multi-gene panels can provide valuable insights by testing a range of genes associated with the suspected condition. Some applied questions I want you to consider when thinking about multi-panel testing are, why may you choose a panel over single gene testing? Why may a client choose a small panel over a large panel? What would be your next testing step if you encountered a total gene deletion on a panel? And what would be your next testing step if you received negative results from a large gene panel? Feel free to pause the video here and jot down your thoughts. Next, let's zoom out to the genomic or book level of clinical genetic testing. First, let's take some time to differentiate between whole genome and whole exome testing. Whole genome sequencing involves analyzing the complete DNA sequence of an individual's genome. This includes both the coding regions, which are the exons, and the non-coding regions, which are the introns and other regulatory elements. By capturing the entire genome, whole genome sequencing provides a comprehensive view of all genetic variations, including single nucleotide variants, copy number variations, and structural variants. This depth of information can be invaluable for complex cases where the genetic basis of a condition is unclear. On the other hand, whole exome sequencing focuses solely on the exons, the approximately 1 to 2% of the genome that actually codes for proteins. Despite the smaller scope, whole exome sequencing is often used because the majority of known disease-causing mutations are found within these coding regions. Whole exome sequencing is generally more cost-effective than whole genome sequencing and can yield significant insights, especially when looking for genetic causes of diseases. However, one limitation of whole exome sequencing is that it does miss variations in non-coding regions, which can also play critical roles in gene regulation and function. This means that while whole exome sequencing can provide a clearer picture in many cases, whole genome sequencing might be necessary for a more thorough understanding of complex genetic issues. In this section, we focus on family testing information as it relates to whole exome and whole genome sequencing. A key takeaway here is that parental trio testing is often the preferred approach in genetic testing. This method involves sequencing the DNA of the affected child along with both biological parents. By comparing the genetic data from all three individuals, we can better identify potential genetic variants that may be inherited from one or both parents. This trio testing strategy enhances our ability to pinpoint de novo mutations, those that occur for the first time in the child and are not present in either parent. Identifying these unique variants can provide valuable insights into the genetic basis of a child's condition. Moreover, parental trio testing can help clarify whether a variant found in the child is inherited or if it arises spontaneously, which is crucial for understanding the inheritance patterns and potential implications for future family planning. This can help us characterize variants of unknown significance. We can ask, do the affected parents have the same variant? In this case, the answer is no, so this family testing is informative. However, in this case, family testing is not informative because there is no evidence of a de novo mutation that would account for the son's phenotype or any variation among the children. Without a clear identification of new mutations, we can't draw meaningful conclusions about genetic inheritance from this family, making the testing less informative. In this case, we cannot rule out the VUS as pathogenic. What are the benefits of whole exome sequencing? One of the key advantages of whole exome sequencing is that it significantly increases the diagnostic yield for Mendelian disorders. This means that more patients receive accurate diagnoses, helping guide treatment decisions. Whole exome sequencing also reduces the time from presentation to diagnosis. For families and patients facing uncertainty about their health, this speed can be crucial. The faster we can pinpoint a genetic condition, the sooner we can implement appropriate care strategies. In addition to time, WES reduces the overall cost of a diagnosis. Traditional genetic testing can be expensive, but whole exome sequencing offers a more comprehensive approach at a lower price point, making it accessible to more patients. Importantly, whole exome sequencing can lead to the discovery of many causative variants. By identifying these variants, we enhance our understanding of the fundamental causes of various diseases. This knowledge not only aids current patients but also contributes to the broader field of genetics and medicine. A unique feature of whole exome sequencing is its phenotype-first approach. This means that before analysis even begins, some genes or variants may be filtered out based on the patient's clinical presentation. This targeted approach streamlines the diagnostic process. Moreover, advances in sequencing technology mean that the turnaround times and prices have dropped considerably. This is great news for patients as it means faster results and less financial strain. Another benefit is that whole exome sequencing focuses on only about 2% of the genome, which means we require less overall sequencing and data storage. This targeted approach not only conserves resources, but also simplifies data management. Lastly, the coding regions of the genome have been characterized to a substantially higher degree. leading to a better chance of interpreting variants meaningfully. This helps clinicians make informed decisions based on the genetic information obtained. While whole exome sequencing is a valuable tool, it's important to recognize its constraints. First, it does not target 100% of the genes in the human genome. In fact, it covers approximately 97% of the exons. This means that some important regions may be overlooked. Additionally, about 10% of exons may not be covered at sufficient levels, which can affect our ability to reliably identify heterozygous variants. This lack of coverage can lead to missed diagnoses, especially in cases where a variant is suspected but not detected. Whole exome sequencing also has limitations in detecting specific types of mutations. These include large rearrangements, structural and copy number variations, such as large deletions or duplications, deep intronic variants, mitochondrial genome mutations, epigenetic factors, mosaic mutations, uniparental disomy, mutations in repetitive or high GC-rich regions, and lastly, mutations in genes with corresponding pseudogenes or other highly homologous sequences. Continuing with the limitations of whole exome sequencing, there are additional challenges to consider. One significant limitation is that whole exome sequencing is unable to interrogate many variants that may be important for controlling gene transcriptional regulation or splicing. These regulatory elements play crucial roles in gene expression, and their absence in exome data can hinder our understanding of certain genetic conditions. There are also technical challenges associated with whole exome sequencing, particularly the need for adequate expertise to generate, analyze, and interpret the sequencing data. A lack of trained personnel can complicate the implementation of whole exome sequencing in clinical settings, potentially affecting diagnostic accuracy. Moreover, whole exome sequencing increases the likelihood of encountering variants of unknown significance, as well as incidental findings. This can lead to confusion and anxiety for patients and families who may not know how to interpret these unexpected results. Ethical issues also arise with exome sequencing. For instance, whole exome sequencing might detect genetic variations that are not related to the disease under investigation. This raises critical questions. What information should be returned to the patient? Should patients be informed about variants that could represent risk factors or causative agents for other diseases? Furthermore, we must consider the evidence required to establish a genetic variance linked to a disease. Establishing causation can be complex and often requires extensive research and clinical data. Finally, there are concerns regarding the ownership, access, and storage of the data generated by whole exome sequencing. Questions arise about whether this data should be retained for possible future use during the patient's lifetime, which introduces additional ethical and privacy considerations. Let's explore when to consider whole exome sequencing and whole genome sequencing. Not every patient fits neatly into a specific phenotypic category or meets the diagnostic criteria for a known condition, making whole exome sequencing a helpful option. It eliminates the need to select or create a custom panel, especially for patients with complex phenotypes affecting multiple organs or systems. Whole exome sequencing and whole genome sequencing are particularly useful when symptoms suggest multiple conditions or when previous genetic tests were inconclusive, offering a broader view to uncover overlapping or hidden disorders. These methods are also valuable when no specific test exists for a suspected condition, providing insights into rare or newly identified disorders. Let's now look at the future considerations for whole genome sequencing. As advancements continue, we anticipate that the difference in cost between exome and whole genome sequencing will diminish. Once we resolve current challenges related to data management and storage, whole genome sequencing is poised to become the method of choice for genetic testing. We are also expecting rapid increases in sequencing technology over the next few years. These advancements will likely enable us to sequence an entire genome at high coverage within a very short time frame. When this becomes a reality, the demand for exome sequencing will likely decline significantly. While whole exome sequencing has its advantages, such as lower costs and faster turnaround times, whole genome sequencing offers many compelling benefits. One of the key advantages is the increased diagnostic yield. With a more comprehensive analysis, whole genome sequencing can uncover variants that whole exome sequencing may miss, enhancing our ability to make accurate diagnoses. Additionally, whole genome sequencing provides greater genomic coverage, capturing a wider range of genetic information. The longer reads generated by whole genome sequencing allow for better determination of copy number variations, rearrangements, and other structural variations. These insights are crucial for understanding complex genetic conditions that can lead to more informed clinical decisions. In summary, as technology advances and costs decrease, whole genome sequencing is likely to emerge as the preferred method for genetic analysis, revolutionizing how we approach genetic testing and diagnosis. As we close this presentation, some applied questions regarding genetic testing I want you to consider are as follows. Why may a provider start the genetic testing process with a CMA over genomic testing? Why may a provider select genomic testing over a large panel? How about a large panel over genomic testing? Finally, what are the best next steps if genomic testing results are negative? This concludes our lecture. Our next lecture will focus on specialized genetic testing. Thanks for watching.